Yeah, all levels are great.
Okay, cool.
Excellent.
Levels are amazing.
Levels. I mean, yeah.
Ooh,
You know what life needs? Quick save. Talking of levels.
Yeah.
Because man, I'd have completely reloaded a couple of things over the last few months.
Checkpoints.
Just in general.
I would say we just have Groundhog Day'd this shit. You know, do a whole bunch of really, really dumb shit.
What's the
and
worst
then
thing
reload.
is, what if the time you do something really horrible is when
You
it
reach the
auto
next
saves?
checkpoint.
It's like, while I finally did that, I killed 20 people, and I know I've done that, and then it just goes ding, and you're like, shit, as the police turn up, and you spend the rest of your life in prison.
You've been playing a lot of Saints Row, huh?
I don't go to prison. They don't get me alive.
All right, so do we actually want to start?
Yeah, I guess we'll start. Okay. Hi, guys. Welcome to Paleo After Dark, our bi-weekly, vaguely science-related podcast. With me today, we're a man down, figuratively speaking. We've got James, me, Randall, and Curtis.
Hello.
Amanda is spending time with her folks at the moment, so she couldn't make it today, but that's okay, because we're going to talk about stuff that she probably doesn't really care about. And you probably won't really care about either.
Oh, that's great, James. Real way to sell it.
Well, I mean, you know, you want to be positive? You fucking talk.
This will probably be the most positive point of the podcast.
It will be, because, to be honest, I'm about to start drinking. And I just don't know if I can keep this level of positivity up right now.
And folks, you should have been listening to what we were talking about while we were reading these papers.
Okay, disclaimers and whatnot beforehand. I think we've already sworn. We're not going to be PG. You guys probably know this by now, but for anyone new coming in, expect various degrees of nudity. Also, we're all studying at the University of Kansas, but the university does in no way endorse or support anything we say. Our opinions are our own, and we're very opinionated. We don't fact-check either. We have read the papers this time, so we're doing better than usual. Aside from that, I think we're good to go.
It was really great last 2 weeks, having Randall with his little notepad.
Oh, crap. Hang on. I'll be right back.
Oh, wait. Okay, so right now James has introduced and then ran away immediately to grab
get
a
his little
notepad.
notepad.
Oh, man. James has made notes?
No.
James is prepared?
No, I'm going to make notes. If I have any good ideas, I need to remember them.
Okay.
This isn't for you. This is for me.
I'm glad we did this.
Apparently, I'm the one keeping the notes for the podcast.
I'm glad we had this moment.
And I now have a time reference to go on.
Oh, okay, good.
So,
Oh, good.
yeah, so, I mean, how is everyone?
Doing fine.
We
Shoveling
shoveled snow.
Yeah.
Yeah, more snow.
Hooray, Kansas. Kansas, which never gets any snow and is usually warm by this point.
What are you talking about? 2 weeks ago, we were grilling.
2 weeks ago, we were grilling. Having trouble with that one, James?
No.
Okay. Come on, it'll pop off. For the record, in case anyone wants to know, he's trying to open a beer bottle, but it has a cork. It's not as obscene as it sounds.
I use my teeth, but then I'll just break my face.
Okay, that might be as obscene as it sounds.
Fucking keep talking.
Okay, well, we'll
All
just
right,
keep
so,
talking while James...
hey!
Hey! Mazel tov.
Oh, now I'm tired. Oh, God. Okay, when are we done?
James, I think it's time for you to get angry.
Well, okay. Well, okay, so Randall, you fucking drew us into this one this week, so do you want to start us off with your first thoughts on... Hang on, do we want to do Dumb or Dumber first?
Dumber?
Let's get to the one that we probably have the most to talk about, because it does have some interesting ideas, but also some potential problems, and let's talk about that. And then the other one, which most of us, let's be honest, skimmed, will just...
It was
This
a
is the level-
64-page PDF.
This is the level of professionalism, people, that we're really hoping you appreciate. Anyway, Randall.
Right, so our first discussion point is molecular timetables.
Okay.
What?
Specifically,
Molecular timetables, 2 a.m. replicate. a.m. 3 a.m. split.
Man, I can't wait till 4 p.m. meiosis, man. I really love that shit.
yeah.
Okay, do you know what the difference between mitosis and meiosis is? Do you know how I was taught this?
Okay.
Okay, so a man is walking through his house and he stubs his toe on the wall and it starts bleeding profusely and he starts shouting out, "Mitosis, mitosis!" He walks into his bedroom and finds his sister having sex with her boyfriend on his bed. He goes, "Meiosis, meiosis!"
So,
Seacrest out.
yeah, that is interesting. That's a good way to put it. Mitosis, or cell division. Meiosis, or the taking of a diploid to a haploid state. For sexual reproduction.
And so what does that have to do with what we're talking about, Randall?
Yeah, what are the molecular timetables, Randall?
Right, so we're
The answer is nothing.
Yeah,
Yeah.
no. It probably has something. No, wait, no, it doesn't.
we have no idea what the hell this is going to
We're talking about molecular clocks today.
Specifically, we're discussing the Rota Stabler et al.
Rota Stabelli.
Stabelli.
Roti Stabler. It's like you can't pronounce fucking genus names or anything.
Nope. It's all Latin to me.
Like you kept getting the one last week wrong.
Well, wait.
Push on!
No, we're not. We are not having
Yeah.
that discussion again. Not without Amanda to punch all of us.
Actually, I think we're
No, this is why we can have this
we're
discussion.
already doing worse this week because Amanda isn't here.
So we're suggesting that this is okay. Yeah.
I mean, I'm wearing my shirt for nothing.
It's true.
Yeah.
It's true. We're not fighting.
No, my shirt says. Can you read it for me?
Why does every day involve a fight with an American?
Whereas Curtis's shirt is Mario on Yoshi, in a purely
Platonic.
That's it. I was going
Yeah,
to say placental.
yes. No. There's no implied
There's
man-dinosaur
no,
relationship.
Purely placental way.
Purely Actually,
And I just hate babies.
Yeah, Randall's shirt just says "I hate babies."
which I think really sums up.
I
Randall's
suspect it was. I
babies.
suspect it was a typo, and I really think they thought nobody had actually put that, so they added a H to the.
I hate babies.
They go down well with fine ale.
You know, an American friend of mine once told me that a fricasseed baby. Jonathan Swift jokes, people. That's what we're going with.
Fine ladies' gloves.
Fine ladies' gloves. Oh, sure, we can give them food and whatnot, but no, have the Irish eat their babies.
Wait, can I refill this beer for 10 cents?
Wait, no, that's no.
That's the deposit on the bottle.
Okay, so recycling deposits on the ball. In Michigan, if you were to send that to recycling, you get 10 cents.
Wow, so I have 10 cents from this beer.
If you could get to Michigan.
Wait, is that a deposit in Kansas, or is that just...
California.
Well, you're not in California.
Michigan.
And Michigan.
HIIA, so it's Hawaii. IA is...
Iowa.
Really?
Yeah,
Yeah.
Iowa.
OR is Oregon. VT is Vermont.
Vermont, yeah.
Isn't that in Canada?
No.
No.
Vermont is a state.
Oh. Okay. ME is
Maine.
Re-
Because MA is Massachusetts. And MI is Michigan. And MN is Montana. So there's really only other one option is ME.
This country is so fucking retarded. CT is
Because we have
Connecticut.
50 states.
MA is, you just told me that one, Massachusetts.
Yeah.
DE is Denver.
Delaware. Denver's a city.
Okay, whatever. And NY is New York.
Yes.
And that's 5 cents. You only get 5 cents in those states.
Yes.
But Michigan, it's 10 cents.
This is why there was a Seinfeld episode about trying to grab random recycling and drive it to Michigan to make money. Because Michigan always gives you 10 cents. And your recycling in New York is only
They
5.
also give you 10 cents per starling pelt, also I've been told. No, Amanda was saying, because it's an invasive species.
Oh, okay, okay, that makes sense. Yeah, there's a few issues. There's a few instances of that across the states where there's like a plant or something where they might pay you a few cents for every plant that you take.
Yeah.
What if you just grew them, and then pulled them up, and...
Then you're being a dick.
Then yeah, I imagine if you kept coming in every single day with like hundreds of them, they might suspect something and stop paying you.
Show us where you're getting all these plants.
Over here. Totally not next to a grow room.
Okay. Oh well, I've learnt things about America.
You have.
What were you saying, Randall?
Oh yeah, there's like a
Right,
paper.
so, Rhoda Stabelli. Right? Booyah.
Guess the nationality.
No, we're not playing this game.
Buongiorno.
Good job.
Okay, so Italian. Nice. Well done.
Are you sure it's not Italian? I know you're American.
The Italians? I speak Italian the best.
Well, you're discussing...
Wait, what are we discussing now?
Do you know what
Is
the
it
funny
like a
thing is?
paper?
Randall's shirt, when it's folded, it just says, I hate babes.
See, I just see hate bays.
We are never going to let you get this out.
We're discussing molecular time trees.
Oh, trees. Okay.
Yeah.
Molecular
Yes,
time trees.
trees.
Where do they grow?
James, just let him
In a
finish.
computer.
They do grow in a computer.
Specifically, how this one might have predicted the early invasion of the land.
Okay, for the record, why the fuck are we so crazy when all of us are sober?
Well, I mean,
There's no estrogen here.
No, is this the problem? Do we need to drink to focus?
I think, I
God.
definitely do, because, I tend to, not that I'm an alcoholic, but I do tend to have at least one drink a day. And I think when I don't have that, I just start to wander. Plus, there's pressure in my brain because I might have an ear infection.
No, that's actually encephalitis.
Well, I mean, I do have a very large head.
Damn it! Why did you do that, Randall? We needed to let that die. That joke needed to just
No,
crush
it's
itself.
not a joke. It's a fact. I mean, we measured our brains in undergrad, and mine is something like 3,300 cubic centimeters.
So is this like the Oliver Cromwell
millimeters.
thing with like the really big head?
No. I mean, it's not too big. I mean, I'm literally just a big dwarf.
I mean, isn't that people who are? I mean,
He's
actually,
very
it's
good over short distances.
very good over short distances. All right, so this is using molecular clocks to try and determine the early colonization times for land.
Yeah, and I think before we get into it, we need
What
someone.
is a molecular clock?
No, I was asking that, Randall. Now you answer it.
See, James thought he could get out of this whole discussion by asking the question. Randall, however, is an asshole
So
That's right.
who hates babies.
So basically, I think we could crowdsource this. With
None
our
of
crowd
us
three.
work on molecular clocks.
I think that should be pointed out,
Yeah.
that this is going to be a very cursory examination of molecular clocks. None of us do work on molecular clocks. We have a general understanding of how they work, but people who really do understand them will probably give you a much more informed impression.
You
So
kind
we
of
could-
had the best description before we started.
We'll read out the hate mail for you when we find out exactly how wrong we are.
Speaking of which, have any of us checked the email?
Oh yeah, speaking of which it might have been online or not, but you can get us at Paleo After Dark, spelled the British way, so PALAEO, After Dark, all one word, at gmail.com. Send us your hate, your love, your indifference, just whatever, or any questions that you want to pose to us, and we can get to them at some point in a mailbag. Anyway, what was that random...
No, I was just pondering as to whether you can really ask people to send you love via email, but continue.
Well,
As long as they're not paying us.
I want to send an anthrax email.
isn't that just a virus?
No, it was an email that said this email contains anthrax.
Then you could also download the white powder.
Okay, good.
Well, no, I thought about putting the white powder on a CD-ROM and putting it into my computer, but I realised that it would just ruin me. It wouldn't hurt anyone else.
Yeah, it would sadly infect you with anthrax.
Well, I mean, it was baby powder.
Okay, well then it would just ruin your
It's
CD
because
drive.
in England there was a ton of anthrax
Also put
letters
them on
going
the
around, but it
no-fly
was
list.
all talcum powder.
Yeah, that happened around here too. Like really early, just after 9/11, there was the media scare on anthrax, and then suddenly there were all these white powder letters.
Do you know? Okay, anthrax is almost eradicated from the wild, but it still grows in Afghanistan.
Yes.
And in some valleys there. And when I was interning at the Natural History Museum in London, it's fascinating. The collections there are amazing, but the real fascination was being in the lunchroom with all the curators and all the doctors. And one of them was saying that his graduate student was currently off ill, and they go, "Oh, why?" And it's like, well, she was doing field work, and she caught anthrax.
Oh man.
So managed to find some real live. What is it, plant? What is it?
It's a fungus, I thought.
fungus, okay.
Yeah.
Yeah.
Or like a spore of something.
So it's
that
basically
you
a
get.
plant.
Yeah. I guess it would be a plant at that point.
Well, I mean, funguses have spores, fungi,
Yeah. I mean,
but I don't care about them.
So wait, you only care if it's a plant? If
No.
anthrax was a fungus,
No,
you would
I-
just dismiss it from your
I don't
own
care
mind?
about plants either.
Okay.
He cares about what we're about to talk about.
He cares about molecular clocks.
So yeah, we should get back onto that.
Yeah, let's talk
So-
about molecular clocks.
So I guess, from my understanding, the main idea of the molecular clock is that if you assume rates of change, you can look at how much changes occur between two organisms and work out when they diverged. Right?
Yeah, so
Yeah.
you're looking at a molecular data set and you're creating a model for gene change through time.
But it's tried to be constrained by the fossil record.
Yeah, so you're
That's
gonna,
when they do it good.
yeah, when you do it well, you have a phylogeny already, and you constrain you sort of pick specific dates. Like I know, or I can imagine based on the data that. Imagine is a bad word. I presume based upon the data that that this particular event has occurred at this point on the tree, and then you use your molecular data, and you typically what you're doing is I guess it's called a soft clock, where you don't have a rigid like strict rate of change through time, but instead it can vary due to the points that you're actually that you're constraining it with.
Mm-hmm.
And so like, I assume due to the fossil record that this has been originated at this point, so I'm going to make sure that in my data that this change happens at this point. And then you run your model and you assume between those calibration points a roughly constant rate of change through time.
And I mean, you need the calibration points, because one thing I got told was
Well, let's back up really quick, just to finish off. So once you have that data, what you're trying to do then is you're looking at your basic calibration points, but you're also looking at all the other changes through time given your model,
Yeah.
and you're saying, oh, well then that suggests that this change happened at this point, and this change happened at this point, and they use that to try and take molecular data, model it into very deep time. We're talking like early Cambrian kind of stuff, and all the way back.
Yeah.
and your calibration points for fossils are your minimum divergence. You know that, say, crown group arthropods occurred by this point in time, so they evolved beforehand, but you know it's not going to be any sooner than the point that you have the fossils for.
Exactly.
You also need, though, to have your maximum divergence point. I got told that when they were doing molecular clock analysis on Nidarians, unless they constrained the age of the Earth, it would suggest that they diverged from each other prior to the formation of the universe.
Well, I mean,
Before time began,
the early
there were Nidarians.
some of the earliest work on molecular clocks, well before even I think soft clock kind of stuff, when it was just a hard fast rate, suggested a protostome deuterostome split, which is like your major split in animals at something like 3.4 billion years.
Yeah.
Yeah.
I mean, early work on these things, and this is, we need to remember actually, this is a relatively recent field.
Yeah.
It's a
Was it the last 20 years?
something like that. It's a new science that has gone through its teething processes, and people are now starting to do more and more with it. And I think the data we're getting out of these things is becoming more and more interesting and more reliable. But I do remember early on they had issues where they'd be using this for phylogenetic purposes to work out the relationships, and not so much on the divergence times early on, but just how things are related to each other. And the way they get the data often is they'll flash freeze animals and then grind them up to get the RNA or whatever they're using out of them. Contamination was a big issue. So I remember there was one of the first results they got was that ticks are nested within mammals because human hair got onto the slide. And stuff like that. And procedures have progressed quite a lot since they started doing this sort of thing.
Well, and I imagine also if you were smashing up ticks, you'd be getting whatever they were eating as well.
This
And
is why
they
you
mostly
shouldn't
eat
pick
mammals.
ticks off yourself and then test them.
Well, I mean, ticks are crazy anyway because they've SEM'd ticks and they survive the vacuum. And they know when the electron beam is on them because they try to get out from underneath it. And it's only if they get hit by the electron beam that they will die a couple of hours to a couple of days later. Otherwise, they're fine. So ticks are... I mean, imagine if they were any bigger. They'd be terrifying.
Yes,
Yeah.
yeah. Although they probably wouldn't have their incredible qualities if they were bigger.
I mean, they might.
They might.
You
So,
don't know.
so you're just suggesting you want to grow giant ticks? and
No, I
take-
don't, because that's terrifying.
Okay, that's terrifying.
It's the brain bug.
It's the brain bug.
So with this paper...
Oh, the paper.
The paper. So the main thing, I guess, to get out of it is, well, come on, guys, help.
Come on, guys, help.
I'm dying here. Look, I'm trying to talk, but so I guess
dying here.
they actually cover quite a lot. They get this analysis, and then they do a lot of things with it, and they say a lot of things with it. The main thing is that this is an analysis that is used. Was it 5 other analyses?
Yeah, it
Yes.
used 5 other data sets to
Yeah.
try to generate its tree.
Yeah, and then what they do is that they use the resulting tree to suggest timings for the colonization of the land, predominantly by arthropod groups.
Yes, it's mostly looking at arthropods. They also throw in onychophorans and nematodes,
Yeah.
so things that are sort of related to them.
Yeah, and I mean it's an interesting paper, and I think that the divergence times for the clades are probably more in line, perhaps, than certain other analyses I've seen. There is a degree of belief. I mean, there's what John Kelly instilled in me when I did biometry, which is the bullshit alarm.
The bullshit alarm.
Which is when you get any results, if it just feels, if it feels crazy, if it's weird, really look at it, because people's bullshit alarms are more conservative, and you should listen to that.
Yeah, it's sort of a continuation of something I really loved. One of the things Luis Gonzalez, our department head at one point, told me is if you have to prove it with statistics, it probably isn't there.
Yeah.
You should be able to see it and then back up what you see by statistics.
Yeah. So in terms of, and there's another paper which I think we might look at briefly, which suggests a far deeper divergence for these things. But this paper suggests that most of the diversification of at least your arthropod groups, we're looking at late Ediacaran, early Cambrian, which I think is not too.
I mean, it has been
that's just fine.
It has been suggested. I mean, that the Cambrian explosion probably has a larger fuse behind it.
Yeah.
And other proxy data like biogeographic evidence from certain arthropod groups suggests that there's at least a bit of a tail earlier than the Cambrian.
Yeah.
Yeah.
But I think the key to that discussion as well is it's called the Cambrian explosion, and that's not about the origination of life, but the sudden diversification of life. And so there could be ephemeral populations back in the Cryogenian for some groups, maybe.
That's definitely true. I mean, I think it's unlikely that we're ever going to get the origination of a group in the fossil record.
One thing I think we should do is we'll throw up a timescale in the.
Yeah.
So that you can, when we're talking Cryogenian and all that, you can get a feel for where we are.
We'll put a link to the GSA timescale.
To the new GSA.
Yeah.
Yeah. So, and I say the focus of this paper is very much arthropods, which suits me just fine, because it's the only group I really feel comfortable talking about with any degree of bullshit authority.
Bullshit authority.
Well, I mean, because I don't know, but I feel like I convince people I do.
Okay.
I mean, there are 2 aspects to the paper. Whenever I look at a molecular analysis like this, there are 2 things. I guess 3 actually. 3 things I look at. One is the topology of the tree, so how things related to each other, because that's primarily my focus of research, and that's what I tend to look at first. The second thing will be the divergence times, and how that feels in terms of rapidity or otherwise of evolution and that sort of thing. And then the third thing will be like the things that they suggested the discussion as to possible methods or causes or stuff like that. And I think all 3 are pertinent for the discussion of this paper.
Yeah.
Yes.
Yeah. So, do we want to start talking about the topology? I mean, I think a lot of this stuff can bleed into greater philosophical questions about the nature of life and evolution in pretty much any of this.
Yeah. So let's start with topology. By and large, so a bit of backstory in terms of arthropods. There was the big linchpin for discussion in the last number of years has been what the major groupings of arthropods are. So, i.e., is it your chelicerates, which are things like spiders and scorpions, and myriapods, which are things like your centipedes, as one group, and then your hexapods, which are things like insects and crustaceans, which are tasty,
of course.
on the other, or are your chelicerates at the bottom, and then you've got a group which is your myriapods, crustaceans, and hexapods.
And so let's just back up. Chelicerates, we're referring to arachnids, scorpions, whip-scaled scorpions, ticks, mites, that sort of thing.
Yeah. Yep.
And your myriapods, you can... Your bugs, basically.
They're very specifically centipedes.
Right, sorry, so centipedes,
and millipedes.
Right, yeah, I don't know, brain fart.
And so what this paper suggests is actually the result that I think is more accurate, which is you've got your chelicerates on one hand, and then your what they term mandibulates on the other, which is, funnily enough, anything with a mandible, and that is your centipedes, insects.
Italians?
Sorry.
And you have the most interesting failures to recall. You usually find something that has the starting letter and you just go.
We were talking about them earlier. My brain just fired
All right,
off.
yeah, no, we were talking about that.
And your crustaceans. And so I think that's an important result, because the Myriaculata, which is the old myriapod plus chelicerate hypothesis, you only ever used to retrieve through molecular analyses. So molecular analysis these days is supporting what we see with morphology, and I think that's a very important result.
And I think as far as phylogenetics is concerned, like the use of all your available data sets is important, because just strictly throwing in molecular data, also throwing in morphological data, and seeing where they corroborate and where they don't corroborate is interesting and important, especially since if you're dealing with the wrong gene, you might end up with a very well-resolved wrong answer.
Yeah. And I mean, this is an important point. A lot of the molecular analyses are done on a single gene or a single loci.
Yes.
Which to me is a single character. But
I mean, they're treating all of the A, T, G, C's as individual characters.
That's fine, but they're not independent. So to me, I think you want to have multiple genes within your analysis. And then people start doing it 3 and 4, and they'll mix RNA and mitochondria and stuff like that.
Yeah.
And that's good, because they all tell you slightly different things. But a lot of early analyses would focus on the one thing that they can sequence, and the point of the paper wouldn't be the tree, it'd be the fact that they can now sequence this thing.
And to be fair, that's because early on with this kind of phylogenetics, it was a miracle to be able to get it done. I mean, sequencing genomes at all.
I mean, because it's not just the fact that you've got to get the data, you've then got to align it, because there'll be bits missing whenever you sequence it, and then you need to figure out where the gaps are and all that sort of thing. So there was a lot of work, most of which these days is pretty well automated, but it's still a lot of computing time goes into formatting the data before you can even start to analyze it.
So I didn't look into the supplemental materials. Do they talk about the various studies that they took this data from?
Let me pull up my wondrous machine of Mac.
Okay, well, while James is booting up his computer, we
okay,
can... Oh, it's up. Oh, well, I don't have to stall for time then. See, this is the problem. I was going to have like a conversation.
Why do you want to do that?
I don't know.
Conversation?
You have to talk to Randall.
Oh, you're right. Amanda's not here.
Yeah, she's the best one. Everyone knows that.
Yeah. Randall just hates babies.
Or eats babies.
Tasty, tasty babies.
Okay, maybe it's a lot harder to find than we thought. Nice job there, James.
They probably say somewhere. Curtis, there's like 142 references.
It's fine. Let's stop that. I was just curious. I didn't know whether or not they ran an analysis themselves or if they assembled based off.
It's all assembled based off
previous studies.
Yeah. Okay.
Yeah. But some of them, I know Rochester Belly has previous trees. So some of the studies will be their own work, but they've supplemented it with other people's studies, which is good because, by and large, you tend to get the same results as your own previous work. So it's good to incorporate other people's work and other people's data into it.
Yeah.
And this is one of the things they say. In order to do this accurately, you need competing hypotheses and you need alternate sources of data in order to get away from the single worker bias that will be there otherwise.
And that's understandable. So then, with the topology, then they run their analysis through molecular clock.
Yeah.
And they get divergence times.
Yeah. And I mean, there's some interesting things in terms of. With the topology, it's like they get crustaceans as being leading to insects rather than their own thing, and that again is something that molecular analyses are starting to bring up, and I think there might be mileage in that might be accurate.
So is that actually suggesting a paraphyletic or a grade crustacean?
Yeah.
Okay.
And considering that would explain a lot as to why there are no pre-Devonian insects just being terrestrial crustaceans.
Yeah, that would make sense.
And then the other thing is the groupings within chelicerates, which are my particular group. There are some oddities there. I think part of this is a long branch issue, which is when your data is so distant from each other that the computer starts drawing connections that are spurious. So there's that, and there's also an odd thing where with molecular data there seems to be a problem by and large, and I'm talking a lot here, so jump at any point, guys.
Okay.
That when you have things that are secondarily reduced, these are often parasitic, so pentastomatids, your ticks, your mites, and then pycnogonids as well for some reason. They're really weird. They will tend to group together, even though they're clearly not related.
So anything that has gone through some sort of character reduction or an effacement will tend to group together?
It's bizarre, but there does seem to be something there where...
I wonder if that means that there's like similar genetic pathways that these things are following, like maybe a deep-seated genetic
or
parallelism.
they just end up throwing out the same DNA.
That could be.
That could be it, yeah.
Interesting.
So yeah. And then, you get their divergence times. And the divergence times seem pretty good to me. You've got a Cambrian origin for most of your major groups, and assuming cladogenesis, which we tend to, the fact that we've got crustaceans in the Cambrian indicates you've got to have myriapods and chelicerates there as well. Or at least the ancestors for those groups. So.
Yeah, so I guess the thing that becomes interesting is then once they get that data, what do they do with it?
They do a lot, don't they, Randall?
Then we get into
we get into the fact that
lots of assumptions.
We get into the fact that sometimes in science, in order to get your paper out there, you need to make a lot of very interesting claims that maybe your data doesn't always back up.
So the main point of the paper is terrestrialization, and that had me...
Terrestrialization based on which three groups?
Your three terrestrial arthropod groups, your chelicerates, your hexapods, and your myriapods.
Yeah, your hexapoda, good old insects.
That was interesting.
Yeah, that's a good word for it.
Because
they also looked at nematoda and onychophorans as well.
Yeah. But because they've assumed that the ancestors, the moment you've got the groups, so the moment you've got your arachnids and chelicerates and your
it was assumed that they were terrestrial.
Like the ancestors to those groups terrestrialized.
Yeah. Okay. So what we're referring to here is they looked at the tree. And they saw, okay, well, this is the first occurrence. We're going to assume, based upon the divergence times, that this is the first occurrence of, let's say, hexapoda. And then they automatically assume that the first occurrence of hexapoda has to be a terrestrial hexapoda. And that's how they sort of said, well, not only is it the first occurrence of hexapoda at this particular time, but boom, this is the time when terrestrialization occurred.
Yeah.
The problem with that is that it's not necessarily perfectly saltationist. I mean, the thing that will give rise to hexapoda probably doesn't have all the characteristics that define hexapoda crown group today.
Mm-hmm. There are other issues too. Assuming that the moment you get the crown group and everything today is terrestrial, that the first bits of the crown group is terrestrial.
Yeah.
I mean, this is slowly getting chipped away. So scorpions, some of the earliest scorpions were thought to be aquatic. And a lot of them are shown to be terrestrial, but there are still some, like the earliest one is found in an offshore environment, and how the fuck did that get there if it's terrestrial? Considering there was no terrestrial influx from anywhere else. You can't assume these things.
It was obviously picked up by a tornado and then dropped into the ocean, and that's why it was there.
And then, or maybe the other way around, maybe all the aquatic things got sucked up and dropped on land.
Everything dried up. The oceans just retreated far too quickly. The scorpions got caught there.
And we don't even know what the ancestors to myriapods are. We have no idea. We know that they're probably in the Cambrian. Don't know what they are. And guess what? All the possibilities for the ancestors, they're aquatic.
Yeah.
Why should I go on land if there's nothing there?
That's the other thing. Why the fuck?
Okay. Let's back this up. So they suggest with their divergence times, and based upon the fact that, again, they're making this assumption, the first occurrence has to be terrestrial, because the modern ones are, which is kind of a bit of uniformitarianism. The present is the key to the past, writ very, very large in this case. And so they say that their divergence times are roughly in line with, and maybe even slightly predate the plants hitting
invasion of the land,
which would mean that you'd get animals invading land.
There was nothing there
well before there was anything for them to eat.
Why would they do that? That is the big question. Why would you even do that? And it's not even like there's a vacant niche to inhabit.
Well, no, it should be fair. There is a vacant niche, and that there is no niche.
There is nothing.
I mean,
it's a Nietzschean niche.
It's what, so that basically the sea was the big hole they had to climb out of?
Yes,
Yeah.
yes, this is Dark Knight Rises. The sea is the big hole.
I mean, yeah, let's take a metaphor and make it an actual thing.
Yeah, let's take a metaphor and make it an actual thing. That's yeah.
I mean, and I mean, you've got a similar thing where you've got the potential for flying insects before there's any reason for anything to fly, but then they find giant phallic fungus that they could have probably jumped off of. So...
Wait, what?
No, do you not know about Prototaxites?
No, I don't.
Okay, so that is a meter-tall penis-shaped fungus that
Okay.
grew up
Yeah.
in the Devonian.
Okay, it just was wondering your fixation on this.
Fantastic phallic fungi.
Yeah, you just really loved the alliteration,
I do.
didn't you?
I do. But okay, so then we say, okay, so even if there are plants, there's not that many of them, why are they there? Well, they then say themselves that these were probably ephemeral, they lived in marginal marine habitats, and guess what that's not?
Yeah.
That's not being terrestrial, that's going onto land sometimes. And
I mean, that would mean Eurypterids could possibly be terrestrial when we have potential evidence that they go onto land, but they don't live there.
But then they say, oh, they could have been eating things that washed up from the tide. Here's a big problem with your aquatic organisms. Do you know how they feed?
Filter feeding.
Most of them use nather bases on the base of the legs. Do you know what you need in order to do that?
Water.
You need to be in suspension. So guess what they weren't doing when they went up onto land? They weren't feeding.
And I think what some of this is getting into is they make some interesting estimates for divergence times. They have this big tree where they've assembled, but they feel it's always the problem is if you just present data, you'll often get ignored. So I think there's probably at least some push to be like, let's try and make the most of the data that we have and really tell a good story. But you end up just telling a lot of stories.
There is nothing wrong with Christ. There is nothing wrong with the discussion. Where it started, and this is not, I'm not being overly critical on the paper. I think it's a very interesting paper that brings up some very good points, and the method they use seems to be pretty good. But when you start claiming that this supports what you're saying, and it's not like it's support, it's just it doesn't conflict with what you're saying. You're starting to stretch it quite a way. I mean, there's this other thing they mention in regards to the rise of crustacea.
Yeah.
Yep.
Which I think is a little bit dubious at best.
Because they have their divergence times for crustaceans, and we know crustaceans are around in the Cambrian. Crown group, or at least upper stem lineage crustaceans are around in the Cambrian. Nothing wrong there. But, and they rely on some disparity work that was done in the 90s for this as well, that as crustaceans get more and more common and seem to inhabit more and more niches, your trilobites and eurypterids appear to shrink in diversity. Now, diversity, bear in mind, is pure...
Not diversity, disparity.
They say diversity.
Oh, they say diversity?
They start with disparity, then switch to diversity because nobody's done disparity analysis on eurypterids.
Okay.
And diversity, by that point, you're looking at taxonomic count, and the question is, what taxonomic level are they working on?
They're probably looking at families.
Yeah. And the thing is, and so what they say is that this means crustaceans outcompeted trilobites and eurypterids, and that's just something you can't test with the data at hand. We don't know if they're in the same geographic regions.
We don't know if they're occupying similar niches, if they're if they're actually eating each other, or they're doing any of that sort of thing.
I hate to say it, we don't know what the trilobite niche was because we don't have their limbs half the time. We don't know what the eurypterid niche was because no one cares. Part of the idea is there's this thing called Angustodontus, which is a mantis shrimp type thing, which has got these raptorial maxillipeds. And when this was first discovered, they only had the spiky bits. They thought it was a eurypterid. The moment that they realised it's a crustacean, they were like, this means crustaceans were moving in on the eurypterid habitat during the Devonian. No, it doesn't. It doesn't mean that. I mean, look at Africa. Look how many predators there are. They do not all directly compete with each other.
No, and that they do just fine, more or less.
And just because even if you get your standard double wedge, which is one group seeming to go extinct as another group diversifies, that's a pattern. You cannot imply causality from that.
No, I mean, you probably, more often than not, you probably have what Gould's referred to with the ships passing through the night, where he was talking about the rise of the bivalves and the falling of the brachiopods, and that for a long time was treated as being like the ultimate example of outcompeting at the end of the Permian. Like, oh well, the bivalves drove the brachiopods to extinction. When if you look at the data, it suggests more that the brachiopods took a massive hit at the Permian and were on the way out because of the extinction, and the bivalves didn't care.
Yeah, and I mean
And that's at least as far as trilobites. What's happening if you look at the evolution of trilobites? You get the trilobites are basically massive speciators. They like to go crazy when they have the time, and then they get massively culled during these extinction events. Yeah. So possibly Cambrian. There's still issues as to whether or not most of the Cambrian stuff just has evolved into Ordovician forms, and Cambrian workers don't talk to anyone outside of the Cambrian. Yeah. That's a major suggestion. But and Ordovician definitely takes a massive hit after their huge rise in the Ordovician. But then they come right back, and they're doing great in the Devonian, and then they get hit again. And it just so happens that the only thing that survived the Devonian mass extinction event was one group.
But there's many, many species of them.
There's tons of them, because that's everywhere. All they do.
Yeah, and I mean, and the difference is, I mean, they actually cite one of my papers through Ipted data, and this is a different matter entirely. They're the first people I've seen that have cited this for things I actually say in the paper.
So you just wanted to champion that one, didn't you?
So, I mean, that's amazing. I mean, there's a whole different discussion there about high-profile papers and people citing them for things that people don't say. And I didn't appreciate this until people started citing my paper for weird things that I have no idea why. But anyway, and in that, with the Eurypterids, throughout the Devonian, they get hit pretty hard. Not actively, but like they just stop speciating. Things just keep going extinct.
Seems to be what a lot of things do in the Devonian.
By the Carboniferous, you're down to two groups. One that is just your bog standard Eurypterid that just lived and did stuff, I suppose, and then the other group, which were these big freshwater things that probably were sweet feeders. So they've lost the big macro predators. So there was a definite shift in what they were doing as a group, but they're still super diverse in terms of species.
Yeah.
So, I mean, you're bundling a lot of things into a single statement that you need probably 4 or 5. There's a titmouse out there.
James is easily distracted.
Anyway,
he likes seeing the tits bounce.
They're birds, Curtis.
I know.
And so there are 4 or 5 different aspects of that data-wise that they're all throwing into one thing and assuming a lot based purely off when things are diverging.
Yeah, and I had something, then I lost it.
Sorry. You'll get it back.
I'll get it back.
I did. That's why I just talked for 20 minutes.
You didn't just talk for 20 minutes. No, and you had important things to say. No, as far as trilobites are concerned, I mean, as you stated, when you're down to that one last thing that just happens to survive, it's still speciose. There's still a bunch of species of this thing, and they're still following that similar trilobite pattern of being very endemic and very high species density. It's just that for whatever reason, the variability that they had to work on has been culled, and that's kind of a pattern you usually expect after mass extinctions. The things that get really badly hit get canalized into one possible area, and then they can either work their way out of it or they can't.
Yeah.
I mean, certainly that seems to be what's happening in the Devonian with your Eurypterids, where you had a bunch of various forms, but then for whatever reason, the stuff, all the diverse stuff got wiped out. You're left with just 2 basic bow plans, and for whatever reason, they couldn't get their way out of those body forms.
And it's the interesting thing about that this is purely the human way of boxing things up. The things that survive are weird. The things that survive are weird, but then they're normal because they're the only things left.
This is what I was actually going to get at, was when you were talking about the whole notion of your bog standard Eurypterids, is there is so much of this interpretation of looking at the modern and seeing crustaceans are everywhere and they're diverse and they're amazing. They must be great at what they do, and everything else in the ocean must have been bad at it because it's dead. And so we're going to look back in the past and see, oh, well, crustaceans obviously did really well here, and coincidentally, coincidentally, the other things are dying out. So that must mean that crustaceans finally came to dominance to show how awesome they are. That's just the pull of your interpretation from the recent.
Well, I mean, this is, again, just storytelling, but what if trilobites were really good at what they did and crustaceans didn't get a look until trilobites just happened to go extinct?
That's another possibility, too.
That's using the exact same data and the exact same amount of interpretation.
Yes.
And it's the exact opposite.
I mean, it's more or less the modern interpretation of the mammals versus dinosaurs debate, right? Like as opposed to the classic idea of the plucky little mammals like beating out the dinosaurs because they were warm-blooded and had milk. They've instead reinterpreted it as us being the small little rodents under feet that just happened to be lucky enough to survive.
Hey, look, there's this giant thing coming at us. I'm going underground now.
Save yourself, mammals. I'll hold off the asteroids.
I love that comic. I really do.
I throw that in all of my talks about extinctions. So.
Well, but it's also, I mean, it's still getting at this point, right?
Yeah.
History is dictated by the winners, and human beings consider themselves to be the winners of evolution, which is definitely not earned, because bacteria is the winner of evolution.
I've got a question for you, Curtis.
Yeah.
So when they're talking about competition, of course, they're not just talking about things eating each other. They're talking about competition for food and space and resources and whatnot. But in terms of trilobites, do you know of anything that would have eaten a trilobite?
There's suggestions that there's some anomalocarids that could have eaten them.
Possibly. Have you seen the paper that came out last month?
I have not.
So they looked at the Cayley and Emu Bay stuff.
Is this the one that suggests there was other trilobites eating trilobites?
The mouth parts are really poorly sclerotized for anomalocarids, and I like, by the way, that you now change how you say that after this podcast.
Anomalocarids?
Yes.
Well, it's because I was doing the icarids as opposed to anomalocarids.
I know, but you've only started doing that since you...
No, because I was referring not just to anomalocarids, the genus, but all anomalocarids.
Anomalocarids?
Anomalocarids, whatever.
Anyway, and I've always said to people, because I look at modern arthropods, and one of the first things they often do after molting is eat their own exuviae so they can get the minerals back. And considering the trilobite bits they get are often in coprolites, how do we know that it wasn't trilobites
eating themselves?
Shells?
Yeah, but there are these things called eoredliciids from the same localities, and the only thing that was big enough to cause... And they've got wounded... trilobites. So we know something was biting these things when they're alive.
This is actually getting into the discussion of the left side of the trilobites being preferentially hit. Yeah.
That's an entire different discussion of shit that gets into nature that shouldn't get into nature.
We'll save that.
But yeah, so then they said that the only thing that was big enough possibly to cause these wounds were other trilobites. And I think that's, considering... If you've got a calcified exoskeleton, the only thing that's going to be able to break that is another calcified exoskeleton. That's an interesting concept.
It'd be weird to think of trilobites just existing on their own separate ecosystem, eating other trilobites.
There is no input for energy, and that's why they went extinct. They eventually just ate themselves
We
to
must
death.
purify the trilobites.
No. There's not a Highlander, one trilobite to rule them all. Wait. If that's the case and it was proetids, no wonder they went extinct.
That's why they fuse up their head shields so they can't be decapitated as easily.
And then they looked upon the world that they had created, the world they had wrought, killed all the other diversity of all the other trilobite species, and went, why do we do this?
And then they realized, shit, I can't do a Scottish accent.
No, you
Wait,
can't do an
what's a
Egyptian
Scottish
accent.
accent?
No, the French guy does the worst Scottish accent.
Right. Yeah. Sorry. I thought you were talking about Sean Connery trying to do an Egyptian
Everyone
accent.
knows he can't do accents. It's like getting Arnold Schwarzenegger to play anyone other than Arnold Schwarzenegger.
No, it was Christopher Lambert's second English-speaking role.
Yes, it was Christopher Lambert's second English-speaking role. Speaking of which, I love him as French Raiden.
No, I love him as French Tarzan.
I never saw him as French Tarzan.
Great Stroke, The Legend of
a
Tarzan. It's his first English-speaking role.
I never saw it.
What was I saying? Oh,
trilobites eating other trilobites.
Okay. The one thing to think about
could be
for all
only
this,
one.
though, is these are often restricted environments. And I don't know entirely what those environments are for those localities. But in the Ordovician of Ohio, there's a Euryptid locality, and there are coprolites, which probably come from Euryptids, filled with bits of smaller Euryptids. But it's a brackish lagoon that was probably sealed off, so they had nothing to eat but each other, and that was probably an unusual scenario. So I wonder how much of that will be you're looking at restricted environments where they had nothing to eat but each other.
There are certain There's certain limb morphologies of trilobites, just the Cambrian stuff, right? That some of them suggest that they could have done some filter feeding. Some of them.
Are you talking about 40s work?
Yeah, I mean...
Okay, let me tell you... No. Moving on. I think it's potentially an argument that we can get into. As far as the general mechanics of a molecular clock, there are some interesting philosophical issues that we can get to as far as the nature of evolution. This is something that Irwin's paper, the first sentence out of Irwin's paper, in fact.
Okay. So we can actually probably...
We can segue. We can talk about both at once.
No. How about we just take a 5-minute break, and then we'll come back, talk about the next paper, and link it into this paper.
Yeah, that works. Hey guys, welcome back. We've just had, I guess, an extended break.
Yeah, 5 minutes turned into 45 minutes, whatever.
Yeah. Randall's
But
now
for
on
you,
to...
it's interesting.
Yeah, well, Randall's now on to the most interesting blood red beer.
Yeah, that is really interesting. An organic cherry fruit ale. Where'd you find that one, Randall?
Mass beverage.
Nice.
Yeah, but I mean, where's it actually from?
England.
No, it's not. They spell flavour wrong.
They're probably just trying to make it so that it's comfortable for the American audience.
Who the fuck is Samuel? Samuel Smith? That's not a fucking English name. Sorry, I don't mean to swear so much.
Smith?
Yes,
How is Smith not an English name? Samuel.
Nobody's called fucking Samuel in England these days. Melbourne Brothers All Saints Brewery, Stamford. Okay, that's actually... Fuck you! Okay, it's actually Stamford, so... Where's this bottled?
Okay.
Soil Association, what? Organic cherry ale. This tells you nothing!
The important things, like the fact that it's bottled in Stamford.
Where's WA?
Washington?
Really?
Yeah.
Washington's got a place called Tukwila in it?
Yeah, Tukwila. Tukwila, Washington. As in, there's also a Walla Walla, Washington as well.
America's so dumb.
All native names.
Then again, this is bottled in a place called Tadcaster.
Yeah, yeah.
But that's a real name, you know.
Tadcaster.
Yeah, well, probably Tadcaster if you're being me. Lancashire Tadcaster.
Lancashire Tad.
So Randall's got a beer which has got the Tudor Rose on it, so I guess it's legit.
Wait. You just spent a minute and a half talking about how this is not real English, and then suddenly...
No. That's not the Tudor Rose. That's the Lancashire Rose from the Lancasters. And it's from Lincolnshire. I'm getting confused. Continue.
Anyway, you just lambasted this for not being legitimately...
They spell flavour wrong. They don't pander to the goddamn Americans.
You know, James, maybe every day wouldn't have a fight with an American if you didn't start fights with Americans.
They shouldn't argue back.
It was called the argument sketch, James.
So talking of things that are disappointing but apparently legit, I guess stuff, papers, things. Do you know what?
You didn't... What the hell was that kind of transition? Speaking of things that are kind of disappointing...
You should have just
what
kept
I'm
recording.
saying right now.
Do you know
We
what?
should have just kept recording.
We
had a fucking transition and lost it.
I might just not care anymore.
No, we care. Speaking of the, so with our previous paper, we were originally discussing before the break, if we can even remember that, about the various methodologies of molecular clocks and what that might mean in a philosophical sense for how you think evolution proceeds. So what we're looking at here with this particular paper that we were discussing previously, the one on arthropods, the rotostaplers,
et al. Used a relaxed molecular clock.
Yes, which means what, Randall?
Armchairs.
You criticize Randall for not contributing, and then you just keep cutting him off. What? Again, starting the fights.
Well, do you know what? If he made me
He's an instigator.
If he made me respect him, maybe I'd listen to what he said.
Randall, make James respect you.
Go El Kabong on your ass.
And you were saying, I'm sorry, I don't mean to be so rude as to make jokes every time you're talking. Even though I am funnier than you.
It's a pretty nice mouth you have there, boy. Make you squeal like a pig.
He's from, the mountain sticks of Nebraska.
Buttfuck Nebraska.
No, there's no bumfuck Nebraska. There's a bumfuck Kansas because there's a road and nothing. And there's signs for towns.
Okay, to be fair, that's because a lot of the towns out in the west were all just railroads.
No, I'm talking
going
about
nowhere.
north of here.
North of here?
Just north of here.
Then again, on the east coast of England, there were no signs for towns on the roads because we took them all down during the war so that if the Germans landed, they wouldn't know where they were going. We never put them back up.
That might be one of those, road projects that could just help stimulate the economy, give somebody a whole bunch of signs and a job.
Or be like Iowa and take all the signs down.
Yeah, so that you can put them back up again.
No, they just took them all down because it made Iowa look better. Quite frankly, with the signs up, it gives you something to look at in Iowa.
Yeah, there's not much in Iowa. Why were they taking them down? They were obscuring the view?
Yeah. So
we drove through Iowa. That was a lot of cornfields.
You drove through Iowa, and then you went, so this is Iowa, huh? And went, yep.
So there's one speed limit sign at the exit of the town. Get on the highway without seeing that sign. You decide to go 70, and it's a 65. You're gonna get your ass pulled over.
Anyway, so Randall, why don't you define for us what a relaxed molecular clock is?
A relaxed molecular clock assumes there is no constant rate of change.
So it's a non-clock-like clock.
Yes. Non-clock-like clock. Imagine every single arm of the clock is spinning at slightly different
speeds. Every branch
speeds.
could, change it.
Have a different
rates.
rate. Yeah.
As opposed to Irwin et al.'s. James is fascinated by a bird now.
James is just fascinated by anything out the window.
Not quite.
Most things out the window.
Wait till he sees the piece of candy we laid down in the hall.
It's right next to the Punji spear trap, James.
Come on, guys.
So
what is a clock-like clock then?
Well, those rates of change are assumed that they are constant throughout every branch.
Okay, good.
So why would you do that?
Good question.
Because you haven't read anything since Darwin.
Okay, yeah, that could be. So if we're getting onto the assumptions that we're going through with this particular type of analysis, with the relaxed molecular clock, we're going post-Simpson. George Gaylord Simpson and his notion of tempo and mode of speciation, basically suggesting that one of paleontology's major contributes to evolutionary theory is that classic steady gradualism from Darwin is not necessarily the best depiction of evolution, since we know that there are certain groups that are probably going to be changing much more rapidly than other groups, and even within a group there might be changes that occur more rapidly during different time frames.
And this actually was the subject of the best talk I saw at GSA this last year, which was about basically when people talk about tempo and mode, they assume the two go hand in hand and that each tempo is a specific mode. And this was about divorcing the two and showing that different modes can sometimes undergo at different rates. That was very interesting, and I think something that there's going to be a lot more interesting research on in the future. So again, we're at baby steps for some of this stuff.
Well, and I think generally speaking, the notion... Darwin embraced gradualism for evolution because Darwin was going for a uniformitarian approach.
He was personal friends with Charles Lyell.
He was personal friends with Charles Lyell, but I think there was also an element of circling the wagons. The other end of the spectrum was catastrophe, which was basically a creationist viewpoint that you create things and that God creates things and then kills them off and then creates new things and kills them off.
And it's really annoying that the evidence actually suggests that there have been occasional catastrophes, isn't it?
So yeah, the fact Darwin needed to downplay sudden occurrences or sudden changes because anything sudden was suggested to just be preying into this catastrophism angle. And so he kind of needed to throw the baby out with the bathwater. He threw out notions of mass extinctions, potentially even stability of species, because that kind of stuff was dramatically affecting his particular agenda of evolution, because even though nowadays we don't see those as being contrary to evolution, at that time it was a catastrophist screed.
So what Darwin essentially said was that the fossil record is imperfect, and anything that doesn't seem to fit this gradual pattern is because it's missing, not because it's not there, but just because it hasn't been preserved. And this is something you said about the previous paper as well, that they sort of throw into that, and then other aspects of the fossil record as well.
Yeah, this is a more modern, I guess, reinterpretation of the fossil record. This is mostly based on vertebrate stuff, if I'm correct, right? The notion that so in the previous paper they talk about potential issues with the fossil record involving the phylogenetics of fossils, and they suggest that based upon some vertebrate studies, that taphonomy and decay could potentially remove important biologically informative information that would then cause your fossil to look more basal than it actually was. This becomes a little bit problematic when you're looking at arthropods, because decay in arthropods is probably not going to really destroy anything, because what you're getting from your arthropods is sclerotized.
I mean, or even if it's and I mean, the worst case scenario, like chelicerate cuticle is not calcified, but do you know what happens when that starts to rot? It becomes very plastic, so at worst, it takes on a shape completely different to what it is. It doesn't look more basal, it just looks weird.
Now, the one argument that I think is potentially important is they do say also with a lot of early Cambrian stuff, there's potential that mineralized things that aren't biological at all could be misinterpreted as biological. I mean, this is even getting back, weirdly enough, to something that they cite as evidence, which maybe isn't, at least it's been refuted, the mandibulates, or the mandibles on those arthropods from the Devonian.
Strudiella.
Yeah, the Strudiella that we talked about in podcast 0, which may not really be there, but they use that as evidence for as what the most basal occurrence.
The first hexapod, and that's where they date the terrestrialization for hexapods. Oddly enough,
yes.
Bear in mind, and this is something else we'll get into. They date the original, the origin for euarthropods as the first occurrence of Rhizophycus, which is a trace fossil.
Yeah.
Yeah.
I mean, yeah, that's problematic in and of itself, because you can't be sure what is actually making the trace. You can guess, but it's not a body fossil, so
yeah.
Best
Can
guesses.
we make this in the lab like we have with stromatolites?
I mean, the whole point of stromatolites is that there's multiple organisms making them, so they're not anything meaningful as a biological unit. And on top of that, biogenic sedimentation can also create them.
Well, yeah, that's what Randall was getting at. Is we
Yeah.
can laboratory create stromatolites, which are trace fossils. Stromatolites, for anyone who doesn't know, they're basically these up and down sort of undulous traces that have been interpreted as that are stacked on top of each other and interpreted as being algal mounds.
Yeah.
But potentially they could just be random.
Yeah, currently
abiogenic.
the only modern occurrence is Sharks Bay in Australia.
But
Yeah.
because we know what creates them, we assume that they've always created them.
Yes. As opposed to one of the most famous ones, which turned out to be a mound of paint from a car factory in England.
Wait, really?
Really?
Yeah.
So what was this one? Tell us the story, Randall.
What color was it?
Well, it was multiple colors, and they went, wow, this is amazing. We actually have the preserved bacteria at different levels. Well, they went into the lab to test it. It was paint.
So where did it come from? Well, I mean, what part of it?
Was this just like a forgery, or was this just an accident?
It was somebody had picked it up, put it in a museum collection, thought it was stromatolite.
Okay.
Well, I mean, given that there is a car workshop near where I live, and the amount of leaching that gets into the local pond from that, I could totally see a chemical build-up. Interesting. So did they have? So they just found it. The collections had no provinciality for it or anything.
I think somebody put it in there in the 70s, and it wasn't tested until 2005.
But was it so it wasn't published on until then? And then they tested it immediately and found out that it was not.
Was published on.
I mean, to be fair,
point.
something can go into a collections and not be published on because
Yeah.
it's either just been curated.
But this is not something then that was out there and then was refuted.
No.
They realised before they published. That's at least give them credit for that.
This
They
is
tested
why
it
the paper
properly.
changed its direction to, we could also create these in the lab.
Okay, well, I mean that's cool, and it's really good for people to test things and then publish negative results.
Interesting. So then they use the fact that we have paint that's preserving these things, and we might be able to create this just in a natural setting without paint.
Yeah.
And then they did it.
Yeah, I have the papers on my computer.
Awesome. So yeah, granted, Strudiella is probably not as dramatic of an example as a stromatolite as far as potentially just being abiogenic, but still.
I mean, there's also the fact that you've got to get it right. I mean, so I know, as I say, I work on chelicerates, And in this paper, the earliest known horseshoe crab is from the Ordovician. It's not the most basal known, which is a different matter entirely. So there is a long, what we call ghost range for horseshoe crabs. So we've got some things occurring very early on that are relatively derived. So that means there's got to be a lot of evolution happening that we haven't seen prior to this.
Quick side note, what is the definition of a ghost range?
What I just said.
Ghost range is when you have a phylogeny that suggests that there's an earlier divergence that you actually see in the record, so that you know these two things had to have been, that there have to have been ancestors that had persisted far earlier into the past.
Yeah. And
Later into the past.
The Rota Stabili paper gets it right that the earliest horseshoe crab is Ordovician. The Irwin paper, which was only published 2 years before this, and the paper that described the Ordovician horseshoe crab was from 2008, which is 3 years before that, gets the date wrong, uses an older date, collaborates it wrong, and so the calicerates are probably underestimated in terms of divergence time. This isn't the first time this has happened. There was a paper by Japrakash and Hoy that was looking at molecular clock divergence rates for calicerates. And by and large, the methods are probably very good. But they didn't look at the fossil record correctly. They said that the earliest scorpion they had was Triassic. And then their molecular clock said, oh, the scorpions diverged in the Devonian. And the big thing, this is great. This means that there is so much history that we're not getting. And they said something like, this is soon after the Cambrian explosion, which is a typical biologist not understanding how geological time works. And said, so this is really important. So it shows that scorpions diverged a long time before we realised. The only problem with that is that the earliest scorpion is from the early Silurian. So their molecular clock underestimated the divergence time drastically, and then they put my advisor and someone else put out a reply with the earliest fossil occurrences and showed that they consistently underestimated the divergence time for these groups. And it just goes to show, even if you're using, I hate your phone, by the way. It's so loud. Even if you are using fossil divergence times, unless you do an exhaustive literature search and get the earliest one right, that can mess up your results. And unless molecular biologists and paleobiologists communicate and work together, this disconnect is going to cause problems.
Yeah. And I mean, this is why the positive points of the previous paper that we were talking about, where they try and take the fossil data as well as they can and use it for calibrating the tree.
I mean, Rota Stabili, I believe, is a molecular scientist. Alison is a paleontologist, and Davide is a molecular paleobiologist for all intents and purposes.
And really, as far as most of these deeper evolutionary studies, a combination of modern data and fossil data is always to be desirable, because you want to try and get the most information that you possibly can. Even if all you're interested in is phylogenetics, trying to get the best example, the best topology for a tree, you want to combine as much data as you can into your analysis.
And I mean, the ghost range issue is also worth bearing in mind, because a lot of molecular clocks have used Weinbergina as the divergence point for Ziphiosurus, because it's phylogenetically the most basal, but it's found in the Devonian, and we know that they're in the Ordovician. So using the phylogenetically most basal is not the it's not the stratigraphically
most
basal.
But if you're using Weinbergina, wouldn't you be using the ghost range for Weinbergina? I mean, you wouldn't just throw it down to the Devonian. You would look at its position basally and say, okay, well, we know it diverged from all of the rest of arthropods at this point in the Cambrian. Let's say
you would hope so, but they don't.
They don't.
No. This is why whenever you see constraint points for time, go into the supplementary material and see what they used, because, as I said, this paper's pretty good, but they used Rusophycus for the origin of arthropods. They got the horseshoe crab right, but if they'd used Weinbergina and suggested it was Devonian, then it's the phylogenetically most basal, but we know that there's about 130 million years of ghost range there.
Yeah.
And this is the thing. Always check the data on these papers. Always look into supplementary material. It's not there because it's not important. It's there because it's very important, and the journal won't let them put it in because of space.
That's often very true. You find it's difficult sometimes to get all the data that you want into your paper, and need to find clever ways to get it through there.
Yeah.
And if make it obtrusively difficult to get the information, then that might be a bad sign for the paper.
Yeah. Well, it's like the Irwin paper. I emailed it to you guys. I was like, why is this 10 megabytes? It's because it's a 4-page science paper with 60 pages of supplementary material.
Yeah.
Lord, yeah. So anyway, if you're assuming, as opposed to a relaxed clock, if you're assuming a strict clock-like clock, a clock, as we've been referring to this as, what are your assumptions then for evolution?
That it's standard and doesn't change.
Yeah. And the rate of change is constant.
And this has interesting implications for recovery from mass extinctions. You'd think that if say, like, take the end Permian, if it's taken 200 odd million years for life to get that diverse, use 94%, why does it then recover so quickly? You'd expect it to take another 200 million years to get that many species back, and that doesn't happen. So what does that tell you about the assumption on the clock?
And I would even say, like, paleontologically speaking, obviously, that there has to have been changes in the rates through time. But even in modern biology, there's been studies that suggest that massive evolutionary change can happen really quickly.
Yeah.
And if that's the case, then the fact that we can see species in the fossil record at all probably suggests something very important.
There's also this disconnect that, and this is another very interesting research paradigm that could be looked into, the disconnect between genotype and phenotype. And what this is where you can undergo a lot of genetic evolution, so a lot of genetic changes, but not change your morphology at all. But very small, simple genetic changes could drastically change your morphology, often by changing how soon or delayed your sexual maturity is, so, you undergo more or less development. And so this is why
changes within Hox genes to affect development time, or just also limb placement, where certain things go in the body.
And this is why I detest the term living fossil. And to take another Calicerae example, people refer to horseshoe crabs as living fossils because from the Ordovician you can recognise the horseshoe crab very easily. They've still undergone 400 million years of molecular evolution. Those things are not the same as they are now. But they've undergone relatively little morphological change.
Well, and getting back to one of the things that we discussed previously before the break, this is a little bit of that annoying pull of the recent, right? We look at this thing and say, "Oh, look, it looks just like what we see today." When we're assuming that we're not really seeing, we're not looking at dinosaurs and saying, "Oh man, these look so much like all the birds and crocodiliomorphs that we see today." When there are lots of similarities, we just overlook them because they look superficially different enough to us.
And I mean, this was a big thing, and I mean, we all have dabbled in the Cambrian. People tend to see weirdness there because you see the differences, you don't see the similarities.
Yeah.
Yes.
And there's this big talk, and it's one of the things in Irwin's paper. They talk a lot about the origin times of the phyla, but what is a phylum? It's simply the basic division, and do you know what? Soon after life evolves, you're going to get the origin of phyla because it's the first divisions.
We're getting into a major philosophical disconnect of people who see phyla as being something meaningful. And then there's phylogeneticists like myself and James who are of the opinion that a phylum is just a name.
Actually, I'd be interested, Randall, how do you someone that deals more with the taphonomy and the methodology, what is your take on the interesting importance assigned to levels of names?
Right, and it's whatever the name is right now. That's what I'm going to call it.
I guess what we're referring to
is... That's the most underwhelming answer I've ever received in my entire life.
Ask an underwhelming question, get an underwhelming answer.
Okay. I guess we don't recognise our own crazy. We think this is more important than it probably actually is.
Let's put it this way. The name might change because, hey, we have some new data here which suggests something else.
The thing is that there's a very specific group of people that don't just see it as a name. They see it as a unmeasured but quantifiable bit of morphospace.
Yeah, it's the origin of an evolutionary unit.
Yeah, can I just for a second? It's an extrapolation of Simpson's notion of Simpson came up with the idea that all of the basic groups are inhabiting some sort of morphospace. which doesn't really like hold water when you start thinking of carnivora like being this big group, yet it also contains a panda, which doesn't really eat meat all that much.
Yeah.
The Simpson believed that things could hop more for space if they tried hard enough.
If they tried hard enough. You really keep jumping until eventually you get up into the air and you fly. Go, birds, go.
Or you become a panda and then you're just fucked.
Or
not, as the case is with pandas.
And for the most part, this whole idea has been debunked. At least this active view of these like bow plans and morphospace. But it lingers in weird ways in paleontology. And in this way, it's very much thinking of phyla not necessarily as just. A modern phylogeneticist would view a phyla as a necessary evil. It's a name that you give so that you can give a division in the history of life some sort of name to recognize it. But where you define that phyla doesn't matter. To very specific people, however, who are kind of still thinking not consciously but potentially subconsciously in the Simpsonian mindset, a phylum is this unquantified but like imagined bit of morphospace, that these things are distinct enough from each other that we would call them a phyla.
See, to me, phyla is just our artificial term for grouping right now.
Exactly.
It could change at any time.
It is the human need to put boxes within boxes.
And I don't actually think, for the most part, that people like Irwin are actually thinking of the phyla as a thing. They're just using that name as a proxy for disparity of morphospace.
Yeah.
But the problem is...
when you start to conflate those two, you get real big problems.
The problem is, if you were to look at crustaceans today, and you look at modern crustaceans, I mean, what the hell? There's so much variety. They're doing so many different things. But if you look at them back in the past, things are more closely related, so then they don't look that dissimilar. They just look weird. You can't quantify morphospace based on the origins because everything's plesiomorphic, very quaint looking, and looks the same as each other.
Yeah.
I have had this problem with Eurypterids. Finding characters for the basal divisions is so hard because they are stumpy, squat, stupid-looking animals with no characters.
As opposed to the later forms, which are kind of cool.
No, they're weird and retarded in different ways. They get very small, centrally positioned eyes, and I'm pretty sure that is universally an indication of shiftiness.
Getting back to this notion of phyla as a placeholder for disparity, as I said, I can understand what you're getting at on some level, but I think it gets conflated in weird ways where people start thinking about how crazy it is that there's so many new phyla in the Cambrian. And that's ridiculous, because that's all. When you're going back to the Cambrian explosion, the reason that there are new phyla is because this is the first occurrence of the major splits.
Yeah.
And in an evolutionary setting, that just means that, oh, look, here's the basal parts of all these other groups.
Well, at least the first occurrence of preservation.
But what they're really talking about, and this is, again, I think the danger of conflating these two issues, and it becomes maybe it's just a weird issue of language, but it becomes problematic when you use specific words that have so many different meanings that you start thinking of things in ways that get you down completely weird paths and maybe off what, again, this is my personal opinion, off what maybe the more interesting question is. What they're actually trying to get at is, why is there this point in the Cambrian where we suddenly get all of this diversity, where we suddenly have all of the main basal bow plans, to use a Simpsonian term, body shapes throughout the history of life?
Yeah. I mean, bow plan itself is an often maligned German word.
Yeah, because it was used by German idealized morphology.
And I wonder how much of that is because people read the German and then you translate it in certain ways. Bear in mind, there was a big problem with evolution, whereas when people became literate in the past, it's often through study of the Bible. So it's difficult to get aside the idea of a force behind something, like a guiding idea that things are going towards something. You can't get away from that because that's how language was built up.
Well, I find it really interesting, for as much as Darwin was desperate to remove Lamarck from the equation, with Lamarck's whole notion of vitalism, like an intrinsic element of the animal to progressively make itself better, he still couldn't get out of Victorian progressiveness. So it's still like competitions making everything better.
Survival of the fittest.
But he's like stuck, because he still has this Victorian mindset of needing to explain that competition is making everything better, and at the same time being like, but evolution is random.
But a lot of his data came from guided human breeding of stock. And what are you doing? You're trying to select for traits that you find are good. And when you've got that as an example, there's always a guiding force towards the perfect animal that you want. And you can't get away from that when you're using those examples.
Yeah.
Yeah.
So getting back to Irwin's paper, Irwin makes some estimates of divergence time, and what conclusions does he come to?
Well, he's using a clock.
A clock. So we're clocking this clock.
And they do have minimum divergence times from fossils, and we can get into that a little bit later. But they suggest a cryogenian origin from the animal kingdom.
For
right?
the last common ancestor of everything.
Right. And considering the last common ancestor of everything is that bit of lightning that hit that phosphate molecule, yeah, okay.
Yeah. 800 million years ago.
Actually, yeah, that's abiogenesis is a whole other can of worms. If you really want to go into when did
That's
abiogenesis
another
happen.
Well, do you
topic.
know what my favourite thing is? I was once told, well, life evolved only once. I'm like, how can you tell? It's like, well, it did. I'm like, well, considering that species didn't mean anything until you get out bacteria, how can you tell?
Yeah.
Yep.
I feel like there's a weird amount of you saying something and then us saying, yeah.
See,
But no, this is just one of those situations where we're all pretty much in agreement that as soon as you're out, you can only identify a species. Plants are very even difficult to some extent.
Fuck.
Okay.
Bloody biologists and their polyploidy trying to. Ugh.
But anyway, typically speaking, most of the time when we're referring to species, in this particular podcast, or probably in most podcasts, we're referring to multicellular animals. Because when you're talking about bacteria, and if you are someone who works on bacteria or plants, God bless you.
Get a better job.
No, they're fun.
No, we
things
need
to
you
look at.
so that I don't have to do it.
Anyway, if you're one of those people who works on this stuff, remember that your concept of what a quote-unquote species is going to be dramatically different because... As I often say, within a bacteria, one mutation, you've got a new individual, a new population, and a new species.
To be fair, they probably have a harder job than we do.
They have a way harder job than we do.
Yeah. Our stuff's dead. Our stuff's not changing anymore.
God damn it, man. We've already said you don't care what a species is. So,
Randall just cares about the name and whether or not he can shoot it with a laser.
You're alive. Take care of that.
okay, so I have a funny, vaguely funny story that I know. They've got a synchrotron CT scanner in Oxford, and of course you can use this. You can use this to synchrotron different fossils and stuff like that. Get a really good idea of the 3D morphology in the rock, so you don't need to try to prep it out and possibly damage it. Well, the chap that runs it was telling me once that one day there's a massive amount of radiation that gets flooded through these tubes when they do it, and they were cleaning them out one day and they found a lizard that had obviously wandered in and then just been cooked.
Ooh.
But it's cooked in a way that the morphology is perfectly preserved. It just died. So he thought, why not? Threw it on the scanner, CT'd it, and now there's a perfectly 3D internal structure of a lizard that just happened to wander in one day.
Who let the lizard out?
Who?
Here's the most interesting thing, right? So this is a tube that floods lethal doses of radiation through it, and somehow a macroscopic reptile found its way in.
To quote Malcolm, life finds a way. Anyway.
Are there any native lizards that can do that?
We've got sand lizards.
Okay.
We've got grass lizards. We're very imaginative people.
We also have house lizards. When they get in the house, we call them that.
It's cold. I'm going someplace warm. And now I'm dead.
And actually, that very well is probably what it was.
That's why
it needed to bask, and it went to a warm thing.
That's why the aliens were living underneath the nuclear reactor.
Wait, is this the X-Files?
No.
No, this is an episode of the X-Files.
Oh, it's also aliens.
Oh, right. No, it is in Aliens. It is.
Yeah.
It is in the movie Aliens, but it's also in the X-Files.
That's because everything likes warmth.
Generally speaking, yeah.
Yeah.
Yeah, especially if you're ectothermic.
Anyway, clock-like clocks.
Clocklike clocks.
So we were discussing...
Good old CC.
Everything changes at the same rate.
Yeah, no, we already discussed that. I'm saying what after that, what happened with his findings? What did he do with his divergence times? You said they made minimal estimates, and then everyone went crazy.
Okay, so and then they suggest a cryogenian. So basically, we're talking global ice ball world where everything was underneath
Snowball Earth.
meters of ice.
Yeah, the Cryogenian is what we refer to as a snowball Earth, where at this point we believe that there would have been ice in the equator, and thus the Earth would have probably been completely covered in
with possible
ice.
few exceptions, such as around volcanic areas.
So
Yes.
this is kind of why people are really interested in drilling those Antarctic lakes.
Well, it's also why people are really interested in Europa. I mean, the
Yeah.
suggestion that potentially life could find a way on a giant ice ball because it might have done it here.
Arthur C. Clarke's 2010 and 2061, The Frog People of Europa.
Anyway, continuing on with the Cryogenian life and etc.
So, did you print this out, Randall?
No.
God damn it!
Because you sent it to me after I left the building.
Oh, that's not my problem.
Why didn't you print this out?
Because I've been at home all week. It's so they stuff and things and do. And to be fair, the paper very rapidly
Everything
devolves.
diversified just before the Cambrian explosion.
And you've got the arthropods, which are the things I care about, earliest Cambrian, possibly Ediacaran divergences, which is kind of congruent with what they say in the previous paper.
Yeah.
Yeah.
But the deeper
Except
level...
this is going way deeper in time.
Well, this is the thing, because what they're doing is taking the explosion out of the Cambrian explosion. They're suggesting everything happened gradually, and because of that, there must be a multi-million-year fuse of slow divergence leading to this sudden preservational window that we call the Cambrian, that we call the Phanerozoic.
I was going to say, it's a little bit...
There's some preservation
There's Ediacara.
beforehand. So, yeah, the Ediacara, and then the Dushanto embryos, and then we
Are
have...
they embryos, though?
Well, we discussed that in arthropods, didn't we?
Are they embryos, though?
All right. So these are these are the embryos that they found in where? Or
China.
China, okay.
And what are they, Precambrian?
Yeah.
Some of them suggest arthropod embryos.
Okay.
Two questions. One, why did you get an embryo preserved but not the egg it was in?
Yeah.
The other question is, a lot of these, that's misrepresented. Some of these are probably real biogenic structures, and I know some of the people that work on these, and they're very good people. Some of the things, though, by some of the research groups, could be early stage diagenesis cleaving of crystals,
Okay.
Yeah.
which looks suspiciously like early stage cleaving of embryos.
Yep.
Yeah, it is somewhat... It's interesting.
It's dubious.
Well, I'm TAing biology and, talking to people about different cleavage patterns and just having to chuckle to myself that basically it's similar issues with biology of crystal cleavage versus embryonic cleavage.
Yeah. And there was a paper just came out in Paleontology looking at some of the Precambrian / Cryogenian sponge spicules, and it suggested that a lot of these could be diagenetic structures. It's the same problem with all the astrobio life, like the things coming from meteorites and stuff like that, and the early life.
The seeding theory.
And the early life stuff.
So this is just to back up. This theory is suggesting that there's a theory in astrobio suggesting that the basic ingredients for life were seeded by comet or asteroid impacts.
Yeah, but a lot of these comets that have supposed life in them, and a lot of the and allegedly a lot of the early life stuff could be intrusion of ferrous minerals and stuff like that look a lot like bacteria, but are just purely diagenetic in structure because of the way that they've fluid filled the sediment.
Yeah.
Yeah.
That I remember that was some of the talk about some of the Martian meteorites that had hit up north, and when they were looking at them, saying, "Oh my God, there's evidence for life," but you're looking at that, and it's actually mostly just due to the impact itself.
Yeah.
So essentially, they're pushing back the origin of life to a point where, as they say in the paper, it's been considered impossible or improbable for life to have occurred. And they say the same thing about the terrestrialization in the terrestrialization paper. There is no reason, or what is the source? What is allowing life to live at this point in time? And there are theories going back a long time that like hydrothermal vents are the origin point for life because it's the only place you can get mineral resources before you end up with a fully functioning atmosphere and good sun.
Good sun. Oh no, you're talking about the solar luminosity issue.
Yeah.
So way back in some of the earlier time periods, even within the Phanerozoic, when we have actual body fossil evidence, it's been suggested that solar luminosity was still very weak. Basically, that the sun has, as it's burning, has been getting hotter throughout the years. That at the very early stages of life, it was probably dramatically cooler on this planet due to the lower luminosity or lower light output of the sun. But that might have been blanketed by abnormally high CO2. Carbon dioxide levels, the greenhouse gas that we're all pretty much worried about at this particular point. At the way back in time, those massive levels of CO2 that we had, which would have caused hyper greenhouses at this particular solar luminosity, were probably the only thing making things tolerable for life early on.
If you think about it, and if you think about the amount of carbon that's locked into deep sea carbonates, there was way more in the system than there is today.
Yes,
Yeah.
substantially.
But
it's okay, we're working at it.
I mean, the most terrifying thing I could think of is if deep sea sediments raised in temperature by 1 degree and it dissolved, everything dies.
Yeah.
Everything dies. And it's a good thing there's like a kilometre of buffer because the deep sea temperatures change very rarely at all.
Very minimally.
Average 5 degrees Celsius, if I remember correctly.
No, because we're talking 1 degree and we're suddenly past the dissolution point and we're all dead.
Yeah.
We're talking very minimal changes, but because it's so deep, there is such a buffer. Bear in mind, nothing after you get out the photic zone, everything's pretty constant throughout most of history.
Yeah.
Except for Jen would probably come and kill me for that,
Which
being
is,
a geochemist.
Yeah, but that is why, for the most part, when people are doing these estimates of temperature in the ocean, they're doing SSTs, sea surface temperatures, because those are photic zone is where most of the life is living anyway, but it's also the thing that's most variable with climate.
And talking of photic zones, that's the interesting thing about the Owen paper, because they're talking about life when there was no photic zone, because there was a thick layer of ice. And considering that's how you get energy in, this is why you have to start talking about hydrothermic vents.
Well, yeah. I mean, that's...
Because this is not a closed system.
But that's been suggested for a long time. I mean, let's be honest. Early life was probably pre-oxic. I mean, the creation, it was most likely anoxic. The creation of photosynthesis and the flooding of oxygen into the environment has been suggested, at least with the banded iron formations, which are these alternating formations of what is presumed to be major carbon deposits, which are probably dead life, some sort of bacteria, and then massive iron beds or rust-colored iron beds, where previously there was no rusting of iron way back before these banded iron formations. It's been suggested that these could be the first examples of life killing itself over and over and over again cyclically.
They built up oxygen.
Yeah, because originally oxygen was a waste product. We've just been really, really lucky at using a very, very dangerous waste product for our own advantages. Free radical oxygen does horrible things to your DNA.
It's the reason you age, right?
That's up and down. There's questions about telomeres on your DNA and the DNA unwinding.
Shortening
There's
of
toxic
telomeres.
issues.
But that's why wine's meant to be good for you, because it absorbs all.
Again, the jury is still out as to
Well,
whether or not.
chocolate's also good for you because of that.
Well, the thing is, here's the thing, okay? Everything is good for you. Or, okay, not everything. Being stabbed is never good for you. But,
Well, I don't know. If
we went back to the Middle Ages, they would suggest that being stabbed was quite good for you because you had too much blood in your system.
Well, remember, it's blood, bile, pus. What was the fourth one?
I feel like we're backing a little bit away.
we're
Anyway,
going to feel like arseholes when we find out in the future that the cure to all diseases is just placing a magnet on your head.
yeah, we are.
Yeah.
All those head-on commercials, that was the cure to every single disease, is whatever the hell is in that head-on.
So hitting yourself in the head with a tube of wax was the cure for
I don't know
everything?
what that is.
Wait, no, putting wax on you is something completely different.
Anyway, let's getting to the notion. So going back in time, that example, the banded iron formations. So most of your life was probably pre-oxygen, and so also probably pre-photosynthesis. So your really early life stuff, it would probably have to be hydrothermal vents, right?
Well, yeah, I mean, there's a lot of focus on extremophiles and extremophilic bacteria, and trying to sequence that and seeing just how divergent that DNA is. The trouble is, extremophiles have been shown to evolve consistently again and again and again.
Yeah, it's a lot of convergence, a lot of things coming back to these habitats because they're available.
Yeah.
And it works.
And considering the big question is considering these vents could shut on and off almost at will. I mean, once they go, how you can't disperse to another one. Your entire lineage is dead. when that goes. And the nearest one is 1,000 kilometers away. You're screwed. And you've got to hope that another extremophile evolves again in order for that, you're just never going to get a lineage that lasts for longer than a single event.
I've not actually looked into how extremophile. I'm sure there's probably dormant stages and possibly dispersal elements to some extremophiles, because I don't think that individual species are only on one vent.
No, I'm sure that's an oversimplification. But
there are a lot of challenges which it would look like, oh, this must be a primitive state, but there's a lot of perhaps very derived characteristics necessary to living in that kind of environment.
Who says that you always live in that environment? I mean, your planktonic stages might be everywhere, and then you just settle in that when you want to breed, or vice versa. You live there as a juvenile, and then as an adult, you piss off.
Screw this place, man. This place is dead.
All there are these goddamn tube worms, and they're lame.
God damn. Man, I'm so much cooler than you. I'm gonna go fucking float around somewhere,
I'm gonna walk, because I'm a crab, bitch.
I'm a crab, bitch.
And then you find that tube and get sucked into it.
So yeah, this paper seems to be trying to undercut all notions, going back to a very strict Darwinian view and undercutting any amount of catastrophism.
I mean, there are a couple of points they raise, and one of them being like, it's interesting that at the Cambrian, after the Cambrian, you start getting more and more disparity. And I thought, this is an interesting point. Disparity is something I'm interested in. There are ways to quantify it. I myself work on certain types of coordinate analysis that you can at least visualize morphospace, even if it's all relative. You can at least visualize within a group. The figure they show is just pictures of different animals.
Because their definition of disparity, again, is just using the Linnaean taxonomic system. And if there are more phyla, there are more disparity. If there are more families, there's more disparity. Because their assumption is that each family and each genus is like a lockstep of X amount of disparity. It's not quantified, but it's just we know a genus level of disparity, and that's good enough.
Well, I mean, they show, for arthropods, they show two species.
And I really don't mean to sound as reductive or straw man as that is, but that's kind of the underlying assumption, is that you can use that as a proxy, and I don't think you can.
Well, I mean, there's an interesting... Well, the thing that struck me as interesting is they show two arthropods in that figure, two species. One is Olenoides serratus, which is your typical best-known Cambrian trilobite.
Yep.
The other one is Morella splendens, which is figured out as being a weird thing that would show that. A lot of phylogenists will tell you that they are actually very closely related. So what they have done is picked one of the closest snapshots you could find for arthropod evolution to show arthropod disparity. And that shows you that our methods of disparity are still at very primitive stages. You need to be very careful when talking about this.
Well, disparity, I think, is a whole other conversation that we can have, because when you're really talking about disparity, it's something that we can understand, like, intrinsically. Like, I can hold up a pen and a pencil, and you go, well, those two things look different. But actually mathematically quantifying how a pen and a pencil are different from each other is a tremendously difficult thing. And that's when we're dealing, like, with completely... I'm gonna get really reductive here. When we're comparing apples to oranges in a literal or a figurative sense, finding the various landmarks to necessarily quantify that is ridiculous.
This is why I think it's very interesting. The key to doing that, one of the keys to doing that would be, like, if all of us in our research group worked together and we did the phylogenetics, we did the principal coordinates, we did the principal components and looked at the landmark stuff, and then we did the ecological niches as well. That all bleeds into disparity. You need to take it all into account. You can't look at one snapshot, and that's all we're doing, because we're mortal beings and we can't do it all by ourselves.
Yeah. I mean, there's the limits of the human mind, I guess, is what I'm trying to comprehend. We can see something and know that that's meaningful. Yeah. We know an apple is dramatically different from an orange, but mathematically quantifying the difference?
Well, the biggest, I think...
it's difficult.
And bear in mind, I'm a big fan of some of Stephen Jay Gould's writings. I think this is one where he screwed the pooch.
Oh, the disparity argument?
If we were to look at then and look at it now, we'd think these are so... that we wouldn't... And it's like, when you try to take the time slice, the main point of the fossil record is that we can see how things go extinct. We've got the time thing, and he was saying if you take that away, it would look so weird, and like...
Wait, what are you referring to? Are you referring to the replaying the tape metaphor?
The wonderful life idea of, if we were to go back then and look at it, we would think these would be the dominant forms.
Well, I mean,
and if we come back now, but those are so weird, and they're so weird when we look at it then. If we look at it now, they look so weird based on
all right,
what we know now.
So giving a little bit of background information with this particular the book Wonderful Life, which if you haven't read it, you should read it. It's a really well written, actually surprisingly fun book on.
Try to pick up the original copy with actual Cambrian things on it. I've got one of the Eurypterids, which one I'm a fan of. Is kind of sad.
Yeah, I have the Eurypter one too, which is really depressing.
Yeah,
but
the 2009 reprint.
But it's a surprisingly entertaining and energetically written book on the redescription of Walcott's original Burgess Shale collections. And anyway, the argument that James is talking about is this notion that that Gould has, where if you were to, we use the modern and we look back in the past and we say, oh, well, obviously, we've got little vertebrates there and those were going to rise to prominence because we're vertebrates and we know that vertebrates are all over the land and we're so great. But if we went back in time, we would probably look and see, well, the thing that looks like it's most likely doing the best is that Anomalocaris over there. Yeah. And that's the thing that dies out.
Yeah,
I mean, granted, when Gould was writing this, what I think Gould overplayed his hand on a little bit isn't so much the replaying the tape argument, but when he said that when he makes
luck of the draw.
He makes an argument, contingency.
Yeah,
contingency basically.
Yeah.
That
I was trying to avoid that word.
Yeah. Well, when I'm talking about replaying the tape, basically he was saying that if we were to go back in time and we were to have things turn out differently, we would just go back and say, oh, well, this is why this happened. That we're using our modern interpretations of what happened to tell stories about why they happened, when chances are it was just luck that certain things survived and certain things didn't. Where I think Gould potentially overplayed his hand is he goes in and says, Anomalocaris is so different, it's probably would have made a new phyla.
Well, he throws the word Bowplan around a lot. And I say, he does this he has another paper on, Spandrel's paper, where he talks...
Spandrel's of San Marcos.
Yeah, and he talks about how there are functional constraints that mean that lead things to go down certain pathways, and that's a very good paper. But again, he uses the term Bowplan, and I think often he had very good points that the language confused.
Well, I think Gould's use of Bowplan comes more from Gould's anarchic personality and desiring to use a whole bunch of terms that had been poo-pooed and thrown out due to their links. I mean, he really loves throwing up Goldschmidt, and I think that's partly because he appreciates a lot of Goldschmidt's science, but also because Goldschmidt was like a pariah.
Yeah. But I mean, Goldschmidt's side, the main Goldschmidt idea that got Goldschmidt basically ostracized was the hopeful monsters thing.
Yeah,
the whole idea that it was experimenting and these weird things bear no relation to what we see today. But and this is the interesting thing. There was a paper came out, I think, a year ago in a Canadian journal, and it was like the next stage. So Wonderful Life documented stage 3 of the understanding of the Cambrian faunas, and then there's a stage 4, and it cycles. So Walcott thought they were all things that you can be explained, and then they're all weird, and then you could maybe explain them, and then they're all weird again. And now we're getting to a stage where we realise they're mostly stem group taxa, and they're weird because they're primitive and they're independently derived in some cases, but they are on the lineages to what we call the phyla because everything works claded genetically.
And this is, as I said, where Gould overplayed his hand. The notion of him going back and saying Anomalocaris is so weird that if it had survived, it would have created its own phyla different from Arthropoda, when realistically it was an arthropod. I mean, it still was an arthropod. We can place it. It's just weird because it's on the stem.
Yeah. Although some people, and this is devil's advocate, some people claim that it's a stem chelicerate, but they're just badly decomposed. I personally don't buy that, but
badly decomposed to the point where they have a pineapple for a mouth?
Oh no! Well, here's the interesting thing. If you ever look at the mouth of an arthropod,
that's true.
And the mandibles cover it. But if you look at the mouth of horseshoe crabs,
it's just a hole.
It's basically an anus on the front. It looks exactly the same, and if you sclerotize it, you get your pineapple ring.
It's true.
And this is the thing. You can homologize these things. They make sense, but if you focus on how they're weird, you're going to think they're completely different.
Well, yeah, and I think we might be getting into a little bit more of how a standard taxonomist would view evolution and how a phylogeneticist would view evolution, based strictly upon the methodology that we need to use.
If you wanted to define a species, you look at the differences.
Yeah, taxonomy only defines species by looking at differences, and phylogeneticists only care about the similarities.
I think the differences are equally important because, do you know what? maybe there's similarities and you've got it wrong.
So anyway, is there any more that we want to talk about Irwin's paper? I mean, there's certainly bigger issues as we discussed. The notion that evolution is perfectly continuous throughout time just it doesn't hold.
Well, I mean, I guess the point of the paper is to say that there's this cryogenian origin, and then they talk about three or four different reasons to why, given this earlier origin, you do not get this fossil record. So they're looking for reasons for the imperfection in the fossil record. So they're being very apologetic, like Darwin was.
Yeah.
But they don't come down on any one side. They say there's this potential preservation issue. There's the fact that they could be very cryptic. There could be the sea chemistry.
Isn't that they make an ecological claim, right? Which they seem to develop the most.
Yeah.
Yes.
What was that? Is that just like that they needed certain like biota to create new niches that would then allow them to colonize new areas?
And that is an argument for. I mean, that ties into argument of why this sun diversification in the Cambrian explosion. And they like to say the development of new niches led to new opportunities for colonization, which led to new opportunities for divergence.
But that would change the rates.
But here's
that implies a change in rates.
Well, exactly. You cannot. I'm sorry. Biology
Lies
does not work
and
with
slander, sir.
Biology does not work with constant rates. And here's the interesting thing. This is why mass extinctions is very important. Mass extinctions at some level reset rates. Rates slow down the older you get as a group. And it sort of resets them. And here's the other interesting thing. You can see this from like the origin of tetrapods. They switch fingers on and off almost at will. So there is a degree of plasticity, and then you undergo developmental hardening, which is a theory that needs a lot more exploration because, I mean, that's a loaded term, and I don't mean every connotation of that.
Yeah, that's been going in and out of vogue for the past 20 years.
Yep. But there is a certain degree of when you originate, there is more randomness, and then as you prune, you canalize.
Getting back again to what we were talking about with mass extinctions and your Eurypterids and your trilobites, massive amount of plasticity, disparity, get locked into certain areas, and then you can't leave. And it's not so much that one's out competing the other; it's just you're stuck.
Yeah, and if you're looking at that as a trend, guess what the Cambrian fits? It's our first occurrence of that trend that we recognise.
And that's, I think, the important take-home point from Wonderful Life. And maybe not so much, as I said, the overplaying of the hand of, like, could have been whole new phyla, but rather that there was a period of large experimentation. And I think the more interesting thing is we have Anomalocaris, or Anomalocarids, from the Devonian.
Yeah.
Yes.
And we know that they continued.
That is an entire... At some point, we need to discuss the evolutionary faunas. We have to.
Okay.
We need to.
We've already poo-pooed a whole bunch of other things that are related to that. So let's not
today, but in the future. And that's a good point. Just because something is considered more basal and becomes obsolete does not mean it becomes extinct.
Yes,
No.
because the things that are basal are no more or less better than anything else. They just are.
Yeah, and Anomalocarids, I mean, recent research is showing that each Anomalocarid had very, very different frontal appendages, very different mouth parts. They were doing very different things. Broad brushstroking them as apex predators is massively underestimating their disparity.
You have the different species, which some of them, the frontal appendages were like a steak knife.
Yeah.
Yeah. You got other ones which might have been filter feeders. You know, it's...
Yeah.
There's a lot going on.
I think this is just something that sort of struck me. I think it becomes more of an issue in Irwin's paper. But do you find it weird that they're using specifically a method that is like consistent anagenetic, like almost through lineage change, on a data set that's all cladogenetic, individual speciation events? I mean, they're basically they're assuming complete and total gradualism.
Well, this is an interesting point of how you visualize these things. When you do phylogenies, you do your tree. There is no way you can do that without inferring a cladogenetic splitting event for speciation.
Yeah.
There are people that believe if you don't have autapomorphies, so unique characters for an animal or a species, that's directly ancestral to the thing it plots next to.
but that's not necessarily true.
No, I've seen. I mean, I've worked on things, horseshoe crabs again, where they've suggested this, and then we now know that the things they're meant to be ancestral to occur millions of years before these things do. So it's an interesting take on it. I mean, bear in mind as well, with their paper, they sort of tread the bizarre line of these things were very disparate, but they all fit within modern groups. They wedge things into it, like they. I don't think it's in this paper. In other papers, they list Sydney as a chelicerate, and it's really not. That is 1940s thinking right there. And it's interesting that the idea that, so basically there was more experimentation, but these all fit into the stems of various phyla, and it's this bizarre mix of old ideas and modern ideas kind of jumbling together, and they definitely have a gradualism bent to it. They seem to think that instead of going outright extinct, species directly evolve into other species.
And I find this weird. Maybe it's a personal bias, but it's almost like this thing is trying to cut paleontology at its own legs. Like all the contributions of paleo as like, mass extinctions and varying rates through time, and the history of life may be slightly discontinuous. It's trying to say no, we're going to assume that Darwin was right all along and ignore all the rest of it, and basically just say paleontology is too imperfect.
I mean, and this might be somewhat unfair. There appears to be a sloppiness in their picking of data points. As I say, they ignore the earliest occurrences of some things and pick things. It's like they did a cursory literature search or relied on their own knowledge and didn't check, and have picked as divergence points things that are hundreds of million years after the earliest occurrence. And this, in fact, it's interesting. If you push it back, that's not going to change if anything, it'll push their divergence back even earlier in time. Yeah. So it doesn't change the point of the paper, but it seems to be suggesting that this is more important than the paleo data.
Potentially. The one thing I will throw out there is inevitably, just inevitably, you're going to miss a reference because we're all human beings and we can't know everything. And even if we're doing our massive data searches, there might be something that we miss.
That's very true, but I would never be too proud to email the person I know worked on the field and ask them.
That's a good point.
And I spend a lot of time
again,
emailing
we
people.
can't make any assumptions as to whether or not that happened.
No, not at all. The other interesting thing, and this is something that comes out of the previous paper as well. Like in the previous paper, they say a lot about trilobites and about the divergence times of trilobites and the life habit of trilobites, and they say a lot about Cambrian things here, which are all extinct and we have no molecular data
That was something that really concerned me about the last one. Is I almost think with the previous paper, they were talking about the trilobite divergence times versus all the all the other crustacean ones. This is what I get for not drinking. all the Cretaceous ones. So I imagine they were just using the divergence times for crustaceans and then just plotting it out with the times for trilobites. Because there's no way you can have any molecular data unless you're modeling it, and then you're modeling based on a model based on God knows what.
Bear in mind, neither of these papers cite the biogeographic studies that suggest a cryogenian origin for arthropods. They ignore that completely.
Point.
True.
And so there is other data that corroborates or appears to corroborate this sort of thing. And this is the thing as well. For some of these papers, they use continental breakups as divergence points on the assumption that you cannot colonize continents, you can only be on them and then split.
Yeah, and we definitely know that dispersal happens.
Yeah.
Like, a lot.
But then this is the thing that strikes me. If you're going to use, how accurately, bear in mind, neither of these use, and I apologize if I'm wrong, neither of these use morphological data. So we're looking only at extant animals and inferring ghost ranges from specific fossils that we've picked and points, and then we're discussing Cambrian animals that we have not included data on at all.
And I'm assuming probably what they had to have done is they had to have placed the fossils into their tree based upon where they think they should go and then run their model off of that.
True. But then you can do your molecular analysis, and now we have programs that you can use morphological data and include that as well, and get the fossils plotting out. And what if a bunch of these things don't fit on any of the phyla? What if they are just stem arthropods, and what you're saying is completely incorrect because you're assuming these things are a group, and they fall out beneath, and the things you're saying are just wrong because you haven't included these things in the analysis at all, and you're just assuming.
I mean, these are a lot of what-if statements that none of us can really answer because
no. Well, we didn't do the studies. We haven't looked into the studies enough. We're not the people that did them.
We just read them.
But I would always. It's interesting you take one set of data, and you infer things that this data does not directly relate to.
Well, and I think this can go into a broader issue that, generally speaking, when you're in science, you write up a paper, you find some data, and you need to have something to go along with that data.
Yeah. I mean, you need a story. You need to, and this is going to sound really sort of cynical.
Yeah.
You need to spin it. You need to have a reason why people should care about what you've done.
There is an element of selling yourself with any sort of paper that you write.
I mean, you are, in science these days, sadly, a brand. You need to show that you can do good research.
I think you might be going a little bit too heavy on the market speak there, but yeah, I get what you're saying.
I mean, if you are known for putting things out of your arse consistently, people aren't going to take you seriously. If you can be shown to be on good things consistently, people are going to believe what you say, even if sometimes you drop the ball.
Well,
it's the same in any field.
Yeah, it's the same in any field. But ideally, in a perfect world, you'd stand up based upon the evidence that you have.
And I think these are two. I should say these are two really good papers that tackle very interesting concepts, and they look at combining fossils and molecular data, which is really important. But there are other papers that have actually combined the two in a single study instead of using the molecular data to infer things from fossils. You can combine the two directly, and that's where the future of this is.
Yeah, and I mean that's the future, I think, of a lot of biological studies in general. As I mentioned previously in the podcast, combining your molecular and your fossil data for even modern phylogenetic studies can give you a much more clear picture of what's going on because you're hopefully going to clear up some of those long branch issues. If there are fossils that can fill in the gaps of how this became this, you're going to get a better topology or pattern on the tree that is more close to the actual evolutionary patterns. So does anybody have anything else to add?
No.
You suggested this, Randall. Come on.
Yeah, come on, Randall.
That's because I'm the pain bringer.
You're the pain bringer?
Come on. Okay, so from your standpoint, as someone that cares less about the relationships of the animals and more about purely their occurrence, how does this idea of a cryogenian fuse interest you as a taphonomist?
There's no toponymy to do back then.
What are you talking about? This is like the ultimate. This is potentially the ultimate taphonomy argument.
Why aren't they there
Why
when they
aren't
were
they
there?
Yeah.
Yeah, that's the argument, right?
Yeah.
Why is it suddenly that we hit the Cambrian and like, man,
There
now
are-
is the time and everything after that to fossilize?
There's two options. Either they weren't there, or there's something that changed in the environment that allowed everything to fossilize.
Do you believe the idea that things started independently skeletonizing because of change in sea chemistry? Because that's been suggested.
It is one of the possibilities.
Do you think it's parsimonious?
I was going to say, are we back to multiple origins for arthropods?
Yeah.
No. This is brilliant. Arthropods have a single origin, but they independently sclerotized about 10 times.
To be fair, I mean,
Trilobite
messy.
cuticle is different to other cuticle. It's fucking calcified.
Trilobite cuticle
True.
is weird. It's the only example, at least that I know of complete calcification.
I mean, you got some
you
calcification in your decapods.
You get it in certain crustaceans, but it's secondary. Like there's still chitin. Like the thing about the trilobite shell is it's pure calcite.
Yeah, I mean, you've got some like aglaspodids, which are a weird group that some people are doing work on, but we're still not entirely sure where they go. They're phosphatic, and you get phosphatic preservation where it's secondary diagenetic phosphatification, but this is like they are building their cuticle out of phosphate. And the interesting thing is, early vertebrates build their bones out of phosphate.
Phosphate, yeah.
So what's going on there?
Was phosphate just more available?
I mean, the weird thing is if you take this out of the biology and look strictly in a chemical sense, chemicals don't care. Like, whatever is most available and whatever is easiest, it just goes for. So if it can slot in, it'll slot in.
Yeah.
So I do wonder if certain chemical pathways in biology are just an example of what was there at the time and making do. Yeah, and then substituting in something later.
So Randall, is there a big chemical shift that you see in the rock record that would allow there to be this big cryptic diversification in the cryogenian?
I have actually not read any papers on it.
I don't know if anyone has done
I
any
don't
work
know
on
if
this.
anyone's done work on it.
But I mean,
I think there might be some.
That's how you'd need to do it, though,
Yeah.
right? I mean, that's the only way. I mean, as I say, you're this following. This is the whole concept is your field, and as far as you know, either no one's done anything on it or it's come up negative.
Yeah. I mean, to be fair,
People could have done something on it and not published because there's no result.
To be fair, just because there's nothing doesn't mean that there's nothing. It just means we have no evidence for anything.
Yeah. And I mean, here's the other thing.
The absence of evidence is not the evidence of absence.
We need to say that in every single podcast.
Yeah. Considering marine life is kind of focused on the continental shelf, if there were no continents because everything's under ice, where would it go? I mean, this is the thing. I mean,
okay. There's a bit of a bias.
Okay.
there.
I'm looking at the present and extrapolating to the
But
past.
no. There's a massive amount of biomass on the ocean floor that we just don't see.
It's mostly mud.
No,
It's mostly
Stuff.
there's weird echinoderms
Microscopic,
running around and microscopic stuff in the muds. Yeah. There is biomass in the ocean. We just don't see it, and we don't. Again, our own personal biases. We don't recognize it.
Yeah. So I mean, I guess the thing I had a thought. And it's gone.
It's okay. It happened to me too. They die. Thoughts die.
Thoughts, I mean, totally, all the time. They just go. I mean, I had a great thought once. One great thought once. Never again.
This was the most important thought of his life.
This
died.
This was actually kind of vaguely relevant as well.
This was relevant?
Yeah, I know.
I thought that was relevant.
Wow. I know. I mean, when have I ever done that? I mean, I'm 1.5 beers down, and I had a relevant thought. But again, how do you know it's relevant? How do you know I just thought it's relevant?
Well,
given
you're
that we didn't have anything relevant to say before we started drinking.
Wait, did you say relevant?
Yes, he didn't have a relevance.
Relevant.
I do find this really funny that Randall started talking after he started drinking.
Well, that's just why we need to make him drink. And I mean, you're not allowed to drink tea at the start of a podcast anymore, Randall, because you've been way more interesting. And bear in mind, you've only had like 3 gulps.
No. He had the bottle and now he refilled.
Okay. Well, I mean,
I've been filling out what was left in the bottle.
You've just been making
still
it
on his first glass.
You've just been making it look like you haven't been drinking by constantly filling it.
Yeah, the bottle was up that maybe there's an inch left in there.
Okay. Well, I think that might about do it, unless you have anything interesting you want to say.
There's a ghost of something there. It's so annoying.
There's a ghost of something? Just grab at it. Just close your eyes and say the first thing that comes to your head.
It might be the ghost of a phylum that used to exist.
The ghost of a phylum.
Actually,
are you sure it's not Alicia's ghost?
One of the interesting biological. Okay, two bad biological jokes, which is how do you work out a population of tigers?
How?
You find a tiger, shoot it, and the population's n-1.
So Tomb Raider.
And then there's also what happens if you find a new phylum?
What?
You kill it and hope no one ever finds out.
And I think on that note, we'll sign off. As always, I'm Kurt.
I'm Randall.
I'm James.
Have a fun one.
I remember whenever we're in bars or something, no one can hear what I'm saying. Or restaurants when there's just a general background noise and people can't hear me and they say speak up and my response is, well, I'm not rude and I don't shout all the time like you guys do.
Randall speaks low so that no one notices.
It's the quiet ones you need to watch.
Until then, eventually, you should have noticed.
You should have noticed, but you didn't notice. And now
I gave you a 5-minute warning.
Yeah.
5-minute warning to what?